Thank you so much. It's a great pleasure to be here in New York again, and to be the first speaker in this very fine meeting. Hopefully I'm going to give you a quick run through of what constitutes the science background for the rest of the morning and into the afternoon. It's good to think of myeloma as sitting there in its niche in the bone marrow in close contact with nurse cells, stroma cells, and the immune system. And as technology has advanced, we're able to look in more detail at the genetics of how these interactions are governed. And the first level of genetic study was really cell culture and metaphase cytogenetics. And the results of those studies taught us really two things. There are two groups of myeloma, the non-hyperdiploid that are characterized by chromosomal translocations, and the hyperdiploid group that's characterized by trisomies. We got a whole new level of detail with spectral karyotyping that focused on chromosome 14 Q32 translocations, the site of the immunoglobulin heavy chain genes. But another level of detail came from these gene mapping chips, where the green indicates loss of heterozygosity and the red gain. So you can see that there is frequently gain of chromosome 1q, a well-known abnormality in myeloma, and loss of chromosome 13. These became important clinically, and we defined an array of important myeloma-specific deletions and gains, and there were these which seemed to be clinically relevant: 1p minus, 17p minus, and 1q plus. And really some of the lessons from these mapping array studies were, one, the importance of the NF-kappa B pathway, which is shown here with the canonical and non-canonical signaling pathways. And it seemed that loss of negative regulators of this pathway were important in terms of copy number, and amplifications and translocations affecting positive regulators. So the NF-kappa B pathway is important. It can be targeted with proteasome inhibitors. The next important pathway that was learned about was this abnormality of the G1S cell cycle checkpoint, with the translocations all deregulating the D group cyclin, or loss of negative regulators of this pathway, such as CDKN2C, and loss of RB. And I've no doubt that Selena will talk about this in more detail later in the morning. And so I've called this the clinically useful myeloma genome, which really is why we look at these things. And so you have the translocations, hyperdiploidy, secondary events, these copy number changes, but then these adverse prognostic lesions, the 3 adverse translocations, and the 3 adverse copy number abnormalities. And this is just to illustrate the point that these are the adverse translocations, which clearly have a worse survival than other cases of myeloma. But there's a lack of sensitivity and specificity about this approach, with some patients still being alive out at 70 months. And so when you look at how these lesions fall together, you can see sometimes you have a single lesion, sometimes 2 lesions, but sometimes you have all 3 lesions. And it's important to assess all of these events, because you have this sort of impact on the clinical outcome. No lesions does very well. If you have 3 lesions, you have a very poor prognosis. And what I would contend is that over the years, we've made significant improvements in the treatment of low risk or standard risk disease, but we've had very little impact up to now on high risk disease. And what we need to do is specific studies in high risk patients at presentation. And in order to understand the biology more, I think we need to model how these genetic events interact with one another. So this is a nice, simple model of the transition from Muggers to plasma cell leukemia, with events that initiate the process and events that lead to progression. And if you try to put some detail on those models, inherited variation can give you insights into the early phases of disease. And if you do GWAS analyses, a number of genes have come out that have been found in the last years, including the ones documented here that are located at these genetic loci. But I think one of the most important findings, shown in this very appropriately named Manhattan plot, is the association with variants on 11q. And these variants are actually located within the cyclin D1 gene. And it's clear that variation in cyclin D1 in terms of whether it's a nuclear transcript or not is important. Again, highlighting the importance of this G1S cell cycle breakpoint transition. The latest level of technology to be applied to this problem is next-generation sequencing. And you can see that as the cost has come down, it's become more applicable in the clinic. But before going on and just looking at the mutations, I think you can use this technology to ask specific questions. And here's this myeloma genome again. But what you can do is generate baits and pull down regions of it that are of interest. So you can pull down the exon, you can pull down the immunoglobulin genes, and you can pull down the region around c-Myc. So just some essential background before saying what the results of those experiments are. As you undergo development through the B-cell system from a B precursor in the bone marrow to a plasma cell, you have to first rearrange your VDJ regions to form a functional B-cell receptor, and then you have to make it fit for purpose via the processes of somatic hypermutation and class switch recombination that are thought to occur in the germinal center. And this is what we thought about the pathogenesis of myeloma. If this is the germinal center here, that in translocated myeloma, the first hit was driven by class switch recombination. These expanded, went to the bone marrow, where there was a second and nth hit, eventually leading to myeloma. But when you use this technology to look at, to clone both sides of the immunoglobulin translocations, you find that not only are class switch recombination events present, but maybe in about 25% of the total, there are aberrant VDJ recombination events which are driving the disease. And I raise this as an interesting possibility because it would fit with the data that sometimes the first hit can actually be in the bone marrow before the cell reaches the germinal center. I think that's an important point that is debatable, but we need to address it further going forth. The other thing, it's easy to be dismissive of c-Myc in myeloma. All cell lines have an abnormality, late clinical samples have an abnormality. And so if you can use this technology to clone the Myc region and see what happens to it in myeloma, it should tell you what's going on at presentation. And the bottom line of this slide is that 20% of the time, new cases with myeloma have a translocation into c-Myc. And those translocations lead to overexpression of c-Myc. So in 20% of the time, myeloma is a Myc disorder. And they seem to associate as well with these 14, 16 translocations, which is very unusual, I believe. So then comes the great joy, the sequencing, or what has really come out of the sequencing. And it's very difficult to put your finger on it, but I think it has actually been useful. And these two articles were published. And this is the distribution of the mutations, where one or two genes are mutated in 10 to 15% of the time. And then there's this amazingly long tail of some genes that are mutated occasionally. And it's difficult to understand that. But it does seem that the RAS pathway is frequently mutated. And the best way to organize this data so you can somehow or other understand it is to think of the molecular hallmarks of myeloma, ranging from immortalization, the G1S abnormality, NF-kappa B, plasma cell differentiation, RNA editing, and epigenetic changes. And if you do it that way, you can superimpose on this the myeloma-specific variants. And ones that were interesting, I think, in the first papers were the abnormality of DIS3 and FAM46C, which highlights RNA editing potentially as being a novel mechanism in myeloma, as well as a whole array of epigenetic genes. Again, it's tempting to say there's no unifying mutation in myeloma, but actually the RAS-MAP kinase pathway is mutated in up to 50% of cases, and BRAF is mutated in 4% of the occasions. And because there are BRAF inhibitors, it gives you the opportunity to ask, is this a driver mutation pathway in myeloma, which is the one question. If you switch it off, are the myeloma plasma cells addicted to signaling through this pathway? And the answer to that is if you use a BRAF inhibitor, I guess it's best illustrated here, but you can see it on the scans, that there's a reduction in the paraprotein. So it is a driver oncogene in myeloma, and the cells do seem to be addicted to it. We can also use the RAS pathways as a way of looking in more detail at the myeloma clone. And these are kernel plots, which look at the density or frequency of mutations. So if every cell carried the same mutation, it would be a straight one peak. If you have this skewed peak, it points you in the direction of there being subclones that are present in less than 100% of cells. And this is the RAS pathway again. And these dotted lines show you the frequency of the mutations. So you can see there are sometimes very small clones that contain mutations, less than 5%, and then sometimes they're present in all of the cells. And this is important for targeted therapy, because what it's telling you is that myeloma is not clonal, as we once thought. There's intraclonal heterogeneity, where all of the cells are related, but slightly different in some ways, with different biological properties. What we're not seeing is interclonal heterogeneity, where although they're all the same and they're spheres, they're different colors with totally different backgrounds. And this brings us to clonal evolution, Darwin and Wallace. And I think you can apply their ideas to understand what happens with the treatment of myeloma and how it develops. These branching evolutionary pathways that they use to explain the evolution of the species are equally important for the treatment and understanding the pathogenesis of myeloma. Because if you compare the intratumoral diversity I showed you to this diversity within these insects, it's very, very similar. And so there are lessons that we should learn. And I try to outline what these lessons are here, that there's competition between subclones for the stem cell niche. The diversity supports evolutionary fitness of the clone. That evolutionary fitness can lead to treatment failure and resistance, and sites of disease may evolve separately. So it's possible you could have a mutation here that isn't present there. And so is this really true? And so I think you need more than just a low frequency from sequencing. So you can take these kernel plots, pick mutations that are present in the NGS, use them to design PCRs that specifically detect the clones, then sort hundreds of cells into single wells, and then use those wells to look and describe the clonal structure. And so what you actually see if you do that is, this is a clustering diagram, and in this case, the evolution was totally linear. In this case, there's branching evolution, as in the Darwinian idea, and in this case, you can see there's parallel evolution, where within this clone, it's developed a KRAS mutation twice that are present in different cells. And here's these kernel plots again, and if you look at Muggus, the diversity is present very early on in the disease process. And so you can build these Darwinian trees where Muggus develops, becomes complex, smoldering myeloma, and then the final levels of the differentiation and evolution are plasma cell leukemia. And another question you can ask with these sort of things is, it's always been unusual to me why you can have smoldering myeloma one day that doesn't have end organ damage, and then it develops end organ damage, is myeloma that needs treating. And these are some paired samples with the kernel plot before and after progression. And you can see in this attempts to show that there's simply a change in the clonal balance that's associated with the progression of the disease. And so this really does focus attention on whether there's a contribution from the microenvironment on disease progression in myeloma. And so these are the types of models that are good to use. So you have, again, MUGA, smoldering myeloma, and myeloma. You get initiating events, and then the immortalized clones are driven forward by the random acquisition of copy number abnormalities, hypomethylation, acquired mutations, which lead myeloma through this process of clonal evolution and increasing genetic diversity. It's clear that myeloma then, therefore, has to be derived from a stem cell, and a stem cell in this context should be self-renewing and proliferative, but it can be genotypically and phenotypically diverse. But if we're to kill myeloma, we have to target these clonal stem cell populations. And so why has myeloma been difficult to treat? And this is a selection of genomes from different cancers showing the frequency of mutations. And here is multiple myeloma in the middle. And if you express it in a simpler way, myeloma has about 35 non-synonymous mutations, acute leukemia 8, and solid tumors many more. So in many ways, myeloma is a good model for treatment and curing solid cancers. Here's the ALL with its non-complex trunk, easy to kill. Myeloma is slightly more complicated and possible to kill. And here's solid tumors represented by this very complicated truncal system. And so these are, again, coming back to these ecosystems, myeloma, extramedullary myeloma, each of these represented by a different stem cell feature. But what happens when you actually treat these cases? So if you target that lesion with linear evolution, all of those clonal cells die. Here, if you target that pink mutation, say, this pink mutation here, what happens? That branch goes, you get a response, but you don't see it because it's only present in 30% of the cells. And so in terms of targeted treatment and where you go with treatment, you should try to kill the roots of the tree and don't prune the tree. So if there are secondary abnormalities, all you will do is reduce the branching on the trees. And so if we come now from that sort of more targeted approach to conventional chemo, this is what you're doing. You're reducing the disease bulk. And in order to reduce the bulk, based on this, you should use combinations of chemo, maximally debulk, and take advantage of the dose effect of melphalan. And this is an interesting observation, I think, that the rate of evolution equals the rate of disease progression. And so is it any wonder then, if you look at paired standard risk cases or high risk between presentation and relapse, that there are more mutations in the high-risk disease at relapse, and suggesting this explains why they progress. And also, the treatment you use can push forward mutation and disease progression. And this is some data from AML, where the mutation pattern at relapse is consistent with them having been caused by the chemotherapy used to induce a remission. So what factors govern the mutation rate or the evolutionary rate? So at the top you have mutation rate. That can be affected by maintenance. The size of the population, it should be reduced to a minimum and monitored in MRD states. And the fitness of the trait to lead to progression of the disease can be targeted. So we have ways of modulating progression. And here's a series of maintenance studies that will be discussed, no doubt, later in the afternoon, supporting the use of maintenance. But you need to think in terms of these models, what is your maintenance treatment doing? And I would contend it's affecting the competition between a good and a porous clone for access to the niche, favoring the outgrowth of indolent, more slow-growing clones. And so, there are lessons you should draw from this for maintenance. You should continue until disease progression. You could alternate agents, utilize the immune system to mop up cells who are missed by the targeted treatment. And just to finish off, we made great progress in the treatment of myeloma, but there's still issues with relapse refractory disease. And so you need to think about what is happening at relapse. So if these are these branching pathways again, You target the myeloma pathway, make a bottleneck, and you have all of these clones which are present. And so where does relapse come from? It could come from the same clone, it could come from a yellow clone, or it could come from a green clone. This is the so-called clonal tiding, where you can have differential sensitivity at relapse. Is there evidence for that? Yes, there is, and this is data from John Keats and Leif Bergseidel, which support that, where 40% of the time the relapse is characterized by a different pattern than the major clone seen at presentation. So you have to assess relapse differently these days, and to conclude, I would say, in terms of biology and treatment of myeloma, nothing makes sense except in the light of evolution. So I'll stop there and thank you for your attention.